So I'm very delighted to introduce Stephan Guttinger, who's been a regular member of the group and has penned a very smart series of essays about viruses as processes. And I'm also very pleased to announce that he has a new job as of a few days ago at Durham University. And you know, I think this article gives sort of this series of interventions gives us some really important framing tools for thinking about the agency of viruses, for thinking about how these processes are situated within other processes. I think it's a really good framework for working across scale. And I think that the last couple of lines of this essay are really, you know, pointing us towards some pretty deep and important ideas. He says the example of viruses presents us with a narrative that does not focus on the idea of intrinsic dispositions, but emphasizes co-production, dynamicity. Am I saying that right? Basically the dynamic nature of viruses and integration. And it opens a perspective on organisms and lifestyle that prioritizes becoming over being, illustrating how virology can challenge and inform the ongoing debate about processes and substance ontologies. So you're doing a lot both to go down into the domain of you know, viral forms of life and these particles that become processes, and then going back out to the philosophy. So I was wondering if you could just start off by elaborating a little bit more, and maybe just that idea of intrinsic dispositions that you're contrasting this to. Like how do you situate the mutant swarm with all these different you know, particles that are sort of the same but kind of different? How does that differentiate itself from the idea of intrinsic dispositions?
Yeah, sure. Thanks. First of all, thanks everyone for joining. I really appreciate that. Second of all, just when you read that, I was like, did I write that? It's one of those like, are these my words? Like, because I didn't reread it that often. It's like, okay, all right. So I wrote that. So this series of blog posts came out. So we had in 2016, I had this paper on viruses as processes together with John Dupre, and we discussed that in April or June, I don't remember, here in the group. And the blog posts were kind of an attempt to go beyond that paper or to think about a future paper. But it was mostly also a reply to a paper that came out in January 2020, which kind of tried to attack process thinking, which is the Christopher Austin paper that I reference in that third blog post. And what was really interesting to me was, because when I wrote the 2016 paper with John, we kind of, yeah, viruses are processes because like you have to think the whole cycle. You can't just look at the particle and get like, you know, what the virus is. It's kind of it's a changing entity. And at the end of that paper, we came kind of to this idea, okay, so it's a cycle and then it intersects with the human cycle, but the virus gets out all the time again, you know, it doesn't come out as a bacterium or as, you know, it kind of, there is something viral to it and that has its program. And kind of, we had an example of like two battles that merge and they can emerge again because they have some sort of cohesion. Anyway, so that was something that in that paper 2016, we tried to think about, but we couldn't really find an answer. And then in January 2020, when the Austin paper came out, he really picked up on that. Like it was, well, clearly, okay, you know, organisms are plastic, they're influenced by the environment, but clearly there is a program that unfolds, and he traces that back to what he calls and what he treats as intrinsic dispositions. And he's defending a dispositional essentialism. Like he's like, we're more, you know, I'm an essentialist, but not like in as rigid a manner. Like dispositional essentialism kind of allows him to deal with dynamicity and kind of some form of plasticity, at least, not dynamicity. But and we kind of, the paper is really well written and all that, and we try to find answers to that. And so I try to think through viruses find a counter, because he worked completely with organisms. He was just like, look at the human body and how it works, and clearly we have a bow plan, and he doesn't say it's in the genome. He kind of, you know, remains like vague, but it's kind of, it's all in there again. The whole, it's an essential, an essentialism in the end. But yeah, I tried to use viruses, and then what I figured was, what we did in the paper in 2016 was focusing on the cycle and change. I wanted to emphasize co-production, because I think that's much more, there is relationism in there, because it's two things that have to be kind of in relation or working together. But I think the co-production narrative struck me as very powerful, and viruses as illustrating them in a very powerful way. With the swarm, there's always kind of, you know, so the virus is like this mutant swarm, mutant cloud. There's still kind of, it's a swarm of entities, you know, it's a swarm of flies, and each fly is its things, with its thing, with its way of behavior. But I think if you dig deeper into the viral cloud metaphor literature, it can do powerful work. I don't think I'm there yet. So this is with the blogs, it's kind of testing the waters, but I think it's very powerful. I wanted to write a paper out of that, but it hasn't happened yet. So maybe it's not that powerful. I don't know. But well, yeah, I'm thinking about those things.
And drawing you out a little bit more on the bow plan, and again, for those who are just joining for the first time, if you have a question, feel free to put it in the chat or just unmute yourself and ask it. But on the bow plan, you know, if it's not genomic, what is it? I remember that word from taking organismal biology class in, you know, 1995, and it's, you know, this ontogeny, often ontogeny recapitulates phylogeny kind of idea, but what is a viral bow plan, or what is the bow plan and the ideas of this other author you're characterizing, if it's not genomic?
I think for him, he doesn't, I don't think he develops it in any coherent way. He's just kind of hand waving, kind of, there is, you know, there's something without being pinned down to how exactly to formulate that. And I mean, to me, the viral bow plan, because I think it is in one of the blog posts that also that process philosophers, and I think I used Rescher, Nicholas Rescher, that they resort to this kind of idea of there's a definite program to each process. There is some plan, you know, some kind of plan in there, and it is something that that is really tricky to explain, because even in a process view, you want, I mean, the stabilities, you don't want to explain them away, because they are relevant. And we had that last week as well with Stephanie's paper, kind of difference matters, and you don't just want kind of this amorphous, everything's related to everything, it's all kind of, and so I struggle with that, of how to, yeah, where to locate any sense of stability. And in a process view, the way we did it, at least, is always coming back to trying to avoid fundamentalism, you know, the underlying plan, and kind of have this, you know, more level playing field, and thinking in terms of any sort of stability as relative, first of all, and as coming out of an intersection of processes. And that's what I thought was so interesting, reading more about the viral behavior. And, you know, I picked out certain dispositions, like we would say, well, RNA viruses are disposed to be, they're error prone. That means they're not always going wrong, but there's a tendency, there's a, you know, they're disposed to go kind of, to include errors in their, as they replicate their sequences. And really picking out that one disposition and saying, okay, let's just look what science is telling us about this. And suddenly you find all the ApoBec3C, whatever, ADAR proteins, RNA editing processing, and it becomes this kind of, you know, as soon as one process, it's not just the enzyme, it's kind of this process, as soon as it's kind of tuned down, you know, the stability, it tips. It's like, it really shows the fragility of that stability that we think the virus has. And And I think that is really kind of that's an important way to go forward, but it's not, I mean, in the blocks, it's not fully developed, and in my head, it's neither, like, it's just the kernel, the seed of kind of where this could go.
I'd like to bring the other reading into play too. So the Cobb and Birch piece from 1985, Liberation of Life, talks about an organism as a society of events, which I think is a really interesting and compelling reframing of what biology might be. So on page 86, it's talking about, and this isn't just life, but things that would be relegated to non-life. From this point of view, an atom is not a substantial entity, but a multiplicity of events interconnected with each other and with other events in a describable pattern. A mouse is a far more extensive society of events, electronic, cellular, organismic, interconnected in far more complex patterns. So I'd love to just hear about events. I mean, should we be thinking like with Karen Barad, like molecular interactions here? What is an event in the corpus of these thinkers, and how are you mobilizing the idea of an event to think about the virus?
I didn't. So I've read a bit of Karen Barad's work, and every time I read it, I was like, oh wow, this is really really strong and useful. But I haven't. I'm not as familiar with her work as I should be, for sure. With Birch and Cobb, I think I didn't, at least in this book, I didn't find the term event is not as developed or as specific probably as Barad would try to develop it, or as I sense she might be using it. For them, it's maybe an event ontology, process ontology is somewhat interchangeable maybe. I might be going too far, but I think they're not as developed in that sense. For them, it's just a process view, is an event view. But yeah, a viral event, a molecular event, I'm not sure. I haven't thought about, I kind of just used it as happenings. So it's not as developed. But the Birch and Cobb piece is, I mean, this is what, 83, I think, or 85, I forgot. But it's 40 years ago, and it picks up stuff that we then repeated in, John's process project. It's all already there, I think, and obviously they build heavily on Whitehead. But the reason I thought I liked their book is also because, like, society, that's a term that Whitehead uses, but we on the project, and me personally, we have never really read Whitehead in any kind of detail. And I think the Barbour piece, the summary or overview piece, kind of, to me at least, shows why. When I read it, I was like, yeah, I think that's the reason why I feel attracted to the process thinking, but to Whitehead's particular way of going about it, I was always a bit like, and I think Birch and Cobb found a really nice way of using some elements and emphasizing them and building them into kind of more modern biology maybe. But I would be super interested in hearing what other people think of, if they've read or if they might be very familiar with Whitehead anyway, but kind of, because to me, kind of, it starts working with like God, and each thing has its own experience, and I'm like, really, is this kind of, and then like, it's just, I get lost in that. And the Birch and Cobb with kind of internal, external relations, kind of just emphasizing this interconnectivity of events or processes, that was what really spoke to me, an ecological model. And that's obviously also kind of in Whitehead, but I don't know, I would be really interested in hearing how people think of how far Whitehead can help us in these kind of debates. Yeah.
Leaving a pregnant pause for others to jump in. I was also interested, like, you're refusing one event, right, which is the jump. So in part 2 of this 3-part blog post, you're talking about, how the jump, which assumes kind of like a particular moment, there's a reified domain of nature and culture to maybe do a gloss. You talk about like the wild and the wet market as this construction, you're more concerned with the flow, and you're bringing, you know, capitalism in. You're bringing, you know, these processes on, yeah, political and economic levels as well. So maybe if you could talk through that a little bit more, and how your thinking emerged. You know, where do you see the distinction between, you know, the dominant narrative, which is about the jump, and this broader process view that takes into account not only molecular process, but political and economic forces?
Yeah, I mean, my thinking always starts from just the molecular level, and kind of what is a molecular process or thing or event, because I come from molecular biology or biochemistry, and so when I encountered, so sorry, I'm just going a quick detour and I'll come back to the wild and the jump, but like when I came onto John's project and kind of process thinking, I was like, always like, yeah, but what about the molecules? Because they always talked about organisms and their interconnectivity, and molecules are kind of, to me, a really hard case, like molecular biology for a process view, because there's so much independence and focus on the sequence. And so that's what really shaped my thinking about process view, that substance view focus so much on the independence of the thing and its containedness, because that's really strong at the molecular level, that you just have the ACE2 receptor. It is, you know, it's, of course, you know, it will wiggle a bit, but, you know, it's just that entity, that floating unit that stays the same if you move it from context to context. And that's what I really struggled with, trying to explain, struggle with this independence thinking. And when I read about the viruses, I mean, this just pops up like that. So with the Quammen pieces and all that, like there is wild, and the viruses just sit there, and his picture of dust being thrown up, and this kind of this passiveness, and our problem is only once we step in, and each thing has its place, and it just is. And there are, you know, kind of, there is danger out there, and it comes to us if we go in there. And it's just this kind of the boundary thinking that was really kind of what interested me, and where I think, you know, again, obviously, you know, if you emphasize relations and dynamicity, those kind of boxes become tricky. At the same time, I think it's just, and this is the one thing, like, do we want to throw away all the boxes? And I quickly mentioned that in Stephanie's group last week, because like Kwame and other people, maybe they work with very strong enemy pictures. Yeah, we shouldn't step in there, they're dangerous for us. But then, and that's another thing that I struggled with on our project. If everything becomes friend and related and kind of part of me, it goes down a very interesting, or maybe also dangerous route, because I then looked at literature from anti-vaccination activists who picked up on that. And it's hard to find those now on Google, because Google is censoring unilateral, just kind of, you type in, you don't find them anymore. You find NHS, NIH at first, and, you know, kind of, which is good, but it's also, anyway. But those web pages, they were really well, you know, they read into the literature, they knew the papers. How they put them together was different, but they were saying, well, scientists are showing influenza has parts of human you know, they're kind of part of us. The whole symbiosis holobiont literature, and they're saying, don't vaccinate. You're killing parts of you. We're more integrated. It's like we're all related. So vaccines are the devil's work, like, you know, biology. And then they would like have, don't listen to me, listen to this Harvard University professor, and they plug in a Google, a YouTube video where some professor talks about how we need our viruses and bacteria, and to go like, you know, don't vaccinate. And I'm like, you want to keep the boxes, but you don't want the boxes to be too rigid. But how do you do that? So that's kind of that's where I'm interested in, this kind of Quammen style of talking about it, but at the same time, yeah.
May I just jump in there? What struck me about your paper was how certain aspects or dispositions, potentialities were realized in the interaction between two different things. Like, you know, you described the process of how this protein of the cell always is kind of changing what is actually transcribed by the virus then later on. And I was wondering, like, how this process of that you're actually referring to, like the conditions in which some interaction happens, influence what is the outcome, like how this, what you're referring to as becoming, has probably also some kind of essentialism in it, like, because there are, you know, dispositions that are realized in the interaction. You know, it's part of what is, well, what is possible, but not everything is possible. Like the virus can't become a human being in the process, but it can become part of the human being. And I'm kind of also struggling with that, thinking about from a, like, because you also have a biochemistry background. So if you think about a virus that that you kind of inhale, and sometimes you become symptomatic of a disease and sometimes you don't. And there are some kind of environmental factors in that as well. You know, like if it's, now it's cold, and in Germany, at least where I'm based, the people becoming sick and severely sick, that number has risen compared to summer. So obviously something in the environment, like the temperatures, probably, that's also known from other viruses, affects how that interacts. So I was wondering, like, how you frame these conditions of interactions with process view and essentialism and the things you negotiate in your papers, which I found really, really good.
Thank you. Thank you. It's a very good point. I mean, this is exactly kind of the struggle for a process view, or for a mature or interesting process view, is making sense of the stabilities and powers that are there and that have some sort of location. And it's like, for instance, with an organism is always the favorite example of process thinkers, or quantum phenomena, but they're so off. You know, people usually like to stick with organisms because that's kind of us and all that. And it's always this example, you know, the organism wouldn't be what it is if the machine weren't running. And it's not the same for a car. You know, the cogwheel stays the same whether it's turning or not. It's just a cogwheel and all that. But look at us, like, you know, our internal constitution, we have to be active in order to be what we are. So becoming before being, activity before stability, and all that kind of stuff. For a virus, the particle goes off and it floats through the air or wherever it is on the surface, and you pick, you know, it's kind of, there's no metabolism. It's not like, it's not really, it has some sort of power, you know, it has a disposition, and it comes together with the other system, and it kind of, that allows it, you know, it gives the enabling conditions. So there is something in there. The way I thought about it is, it's you move away. So for instance, when you have the virus hit a human body, and in some people disease breaks out, in other it doesn't, it depends on certain conditions. Maybe there's a disposition, it needs certain conditions to be activated, to be realized. But then that, again, the narrative kind of makes, well, you have this thing A, you have this thing B, and they somehow meet. For me, the thinking would be then more historical and more trans-temporal. And that's, again, very interesting, I think, what people have found when they look at RNA editing systems, that the virus in that sense is not naive. The cell is not encountering the virus, that they're part of like, it's like we have to think them as one system that has phases where they're not co-localized, but you could treat them still as kind of contained as one process. I mean, that's where the language breaks down and where it becomes really hard. But what I mean to say is that any virus that hits us hits a cell that has been shaped by previous viral interactions. So there were like these papers that showed like that 30% of conserved proteins in humans show signs of selective pressure by previous through viral interactions, and at the same time viral gene sequences or genome sequences show signs of previous interactions with RNA editing systems. And there are papers now coming out, I just found one or two, that show that coronaviruses do have patterns or signatures of previous encounters with RNA editing systems. So endemic coronaviruses have fewer of the recognition patterns that human RNA editing enzymes need to kind of target them. Whereas in zoonotic kind of viruses, they found less of that. They find more of these patterns still present. So whatever kind of virus hits us, it's always kind of, it's a co-produced, like the abilities it has, its dispositions have already been shaped. So they're not its dispositions, you know what I mean? It's kind of, it's already, it's being co-produced, but not like in the here and now, like we think of the organism, like right now, all these things working together. It's kind of a more temporally extended, less localized co-production. But then how do you express that? It's like, it's a tricky one, but it's a very good point. It's one I struggle with as well.
Can I jump in?
Please.
I'm just thinking off my feet here, but there's something about this question of where to locate any kind of stability. And you mentioned that a while back when thinking about process thinking, how to actually conceptualize where any kind of stability is located, which I think is a huge challenge for process thinking, because clearly there is some tendency to go a particular way in many biological phenomena. And I was just thinking now, would one possibility for framing that being locating the stability in the historicity, right? Like locating the stability in the sort of in the temporal history of this particular phenomenon over very long periods of evolutionary time. So even the stability is a kind of process, but it's a process that we experience as happening, as being sort of law, because it's like this very, very strong habit developed over very long developed and shaped over long periods of time. Yeah, that's just a kind of, I'm just thinking off my feet.
No, it's a very good point. I think the habit kind of and the memory comes in as well, kind of, but then you kind of, the viruses that are floating around now kind of carry a memory that had developed a habit which has been shaped by the habits of other processes. I think that's a very good point. One thing that I want, just when Kevin's question, the constraints, not everything happens. The virus will not just, there's certain limits. And I think, I forgot to mention that, but that's one other thing that I think is very interesting. Energetics, I think the constraints that we observe, and this notion, not everything can happen, that is very true. But that also happens at a non-biological level, if that, kind of at a purely, whatever you want to call it, just kind of a stone or matter. And I think energetics and its role for a non-substance thinking has not been considered enough, probably. And so there's interesting work by Ostwald, Nobel Prize winner in physical chemistry, I think, a really towering figure in chemistry between 1870, 1910. And he wrote a lot about energetics, and he used that against substance thinking. He didn't know about Whitehead, that was before Whitehead, and he really inspired Bogdanov, Lenin's big foe. And anyway, that turned into it's a whole own debate. But the energetics thinking then went away. I don't know many people who use Ostwald, and he went into certain directions which are kind of, anyway, I don't know how well developed that really was in the end. But I think thinking about flows of energy in a more disembodied sense as shaping and forming stabilities and phase transitions, and that is often ignored in talking about molecules, organisms, and so on. I think there's a lot in there that could help shape our thinking about constraints and habits and certain types of flows that seem quite non-plastic or set. So, but again, that's not, I haven't developed that. If anyone has read Ostwald and thought about energetics, because obviously that is in the Anthropocene discussion, that's a big, energy flows and circulation is a big topic as well. But I think that's for me, that's more interesting than going into a hardcore Whiteheadian way, but maybe I'm just too ignorant of Whitehead. So if anyone here has like a proper Whitehead bend in their thinking or knowledge, I would be interested in hearing what's the most powerful concept. So yeah, I would be interested in hearing that.
One really powerful moment in your paper was when you write, what might look like a stable thing is in fact a slow process, and it is from change that stability flows. And this applies clearly to biological organisms. And then you quote Dupre and Dan Nicholson, as process and unlike things or substances, organisms have to undergo constant change to be the entities that they are. And I'm wondering if we think with, so Anna Singh has in the Feral Atlas some new ideas about velocity and acceleration. And maybe one of the things that, as we're thinking about how viruses interact with human bodies or other organisms, but also capitalist assemblages, transportation infrastructures, if it's the speed of the process that is precisely one of the things that needs to be considered. So maybe how do you, if we're all processes, and if the human form is a slower process than a viral form, and we could talk about other even much slower processes than the human. I guess the question is just, how do you think about speed, velocity, intensity, and acceleration amidst all these processes?
It's a super good point. That's extremely central to the quasi-species viral cloud thinking. There are these because people who bring up that picture of mutant cloud or a quasi-species, they are trying to understand how do we have to think about the evolution of viruses. That's one of the key questions they're interested in. And what changes if we adopt this kind of cloud picture? And one thing that is really central there is exactly speed and slowing down and speeding up certain processes, and intensities as well, which kind of goes into energetics to some degree, at least, kind of, not power in terms of dispositions, but literally kind of the intensity, the power of a process. And RNA viruses are exactly at the core of this kind of mutant cloud thinking because of their propensity, of the system's propensity to mutate. And the thing is that they have these pictures, and I read this the other day about survival and which one is the most powerful, which comes out on top. It's the survival of the flattest, not the survival of the fittest. And it's a fascinating, I don't think I got all the nuances of that picture, but basically the idea is you have this balance of processes, and so you have a fast replicator, producing, it's just very quick, and you have a slower replicator, and you have a fast replicator with a slow mutation rate, for instance, they can reach a very high peak in a fitness landscape. They can, you know, they replicate fast and they can reach a point which is really, really fit, where their fitness is maximized. But if you had that same thing and you have an increased mutation rate, because it's such a slim, like you have a sequence landscape, like the possible sequences, and anyway, one of them is like really fit. But as soon as one mutation, because it's so thin, moving slightly over in the sequence space just massively drops your fitness. So that particular fast replicator can't take too much of a mutation rate because it will just fall apart. Whereas you have slow replicators they have overall a much flatter fitness, like they have different variants and they're not super fit, but they're kind of fit, you know, and so they can take much more mutation. And so they're much broader in the sequence landscape. Anyway, and then they have more opportunity to adapt. And anyway, the thinking is that it's all about this balance between fast replication, slow mutation rate, high mutation rate. And it's really just kind of getting, and that's why I thought kind of fitness and kind of the replication and the error thinking, the error in thinking about errors in replication is so powerful, because it shows it's really all about the balancing of these different processes. And then there comes in lethal mutagenesis, or the, what do they call it? The error catastrophe, because like the idea is these viruses are just like they're on the, you know, it's like a drunk driver who's just like on the edge of kind of not going off the road, and it's really crazy. And that's kind of where viruses are. And if you just increase it a bit more, they just implode and they produce too many mutants that are not viable, and just the system breaks down. It's like this just teetering on the brink of collapse, and that makes them so powerful. But then the cell, some people have shown that if you make the virus less error prone, more trust, you know, more reliable, it dies. So, you know, that they really, to be powerful, they rely, it's a funny, yeah. So the acceleration and intensity are key, I think, to this type of thinking, which then also feeds into thinking about jumps and spreading of viruses across the globe. And I mean, I guess it's a trivial point, but the traveling and all these intensities are frequencies. But One Health people have written about that for a long time. I'm intrigued by the One Health literature, because sometimes it's very congenial to what I would think, and kind of, oh yeah, great. And sometimes it dips off into this kind of substance independence, the dust and all that. So I'm intrigued by I don't know, it's a huge literature by now, so I don't know all the people writing on this and all that. So if anyone here has, I'm sure there are people here who have looked really closely or work with One Health, and would be interested in hearing their thoughts on the power of current One Health approach. There will be many different ones, but yeah.
Can I jump in? And apologies for the small erotic distraction.
I think I'm the only one who noticed.
This is morning here. I was interested by how your philosophy of process allows you to criticize the idea of wide reservoirs and spillover, which is also at the center of One Health's reasonings. And I wanted to ask you how the philosophy process is compatible with bioinformatic and views of phylogenetic trees. Is there enough stability in this process to make space for a kind of mapping of the differences with genomics or metagenomics? I mean, is it a form of representing processes that should be criticized, or is it something that allows to see precisely these small processes?
Yeah, it's a good question. I think the process view, I mean, I don't want to talk for other people here, but the process view is usually that the tree the tree would have to be drawn very differently with like cross connections, and biologists are doing that as well. They, you know, it's not, they're not sticking to one tree image rigidly and all that. So, but it's more, I think the fear is always for the process thinker, maybe, again, I'm generalizing, but is reification, kind of presenting, like not emphasizing the dynamicity and the relative stability of these things, and kind of the way a certain depiction, a certain tree picking out certain connections depends on interests, which shape what we measure, how much we measure, at what frequency we measure, which then shapes the dynamics that we can actually see in the system, and so on. And so I think there's just, I think in process thinking, there's still a place for tree and phylogeny and kind of that approach. They would just try and mute it and contextualize it as much as possible, maybe, even though biologists will be doing that. There's always kind of this kind of thinking that process philosophy comes from the outside and teaches biology, as like, you know, kind of informs them or kind of reshapes how biology is done. I think I was always quite critical of that, because in the previous project that there was an element to that kind of, we need to tell people how biological systems really work and they're dynamic. But I think most practicing biologists, they can't evade that, because everything falls apart. You know, it's kind of, it's just the dynamicity is the first thing you encounter. You put everything on ice and on buffers because you don't want it to change. So you just know any sort of stability you want to see, you have to fight for. And people are highly aware of how they're, you know, how their sampling might bias give them a certain picture. And I mean, it's interesting the way they're trying to understand SARS-CoV-2, trying to find some papers on the cloud, the quasi-species nature of that, and it's all still early days and the sampling is not developed enough. What kind of picture are we really drawing? And yeah, so I'm curious to see what comes out of that. But yeah, process thinking and trees, I think, has a place, but in a different way of reading it, if that answers the question, maybe.
I thought I might just offer another iconic image of SARS-CoV-2 that's very different from the particle. So I'm just going to share my screen. So this is can everyone see it? Can everyone see this picture here? So this is the RNA polymerase, the bit of SARS-CoV-2 that's making more of itself. And yeah, it's a model of folded proteins captured at a static, it's rotating in space, but the real lively action is happening as new strands of RNA are being generated, and it's basically what the machine. And here I'm defaulting to these metaphors that are very Cartesian, but yeah, this is an alternative image of what kind of a particular part of the essence is, but then it's processes happening parallel. There's other images of all these other enzymes that are doing other things inside of the cell at the same time that this is happening. So I thought I'd just share a gleaning from ethnographic fieldwork among the virologists who are trying to characterize not the thing, but all the distributed processes of SARS-CoV-2.
I think the enzyme bit is interesting, because the virus case is so interesting to me, because it's, in the end, it's just an assembly of molecules. There is no metabolism inside the viral particle, or at least, no, there's, well, giant viruses, maybe they have the, yeah, there's no sharp, but for the most viruses that we look at here and the SARS-CoV-2, there is no metabolism. It's just a bunch of molecules. And that is where the machine talking extremely quickly comes in, and you just have this particular fold, and if the salt condition, if the pH is right, it has this shape. If pH switches, maybe, it changes shape, but this is coded into the sequence. And that for me is, again, one of the hard cases for a process view. And I tried in other work of mine, I tried to think through that, because there's a lot of interesting work in protein biology, which again goes to clouds, actually. They intrinsically disordered proteins, and that they're constantly changing shape, and that this change of shape is actually needed for their functioning. Super fascinating stuff, like proteins that are phosphorylated and that interact. Usually we have this idea, you add a phosphate and then it has all the properties it needs to bind to a targeting site. But for some of these proteins, a SIC1 is a yeast protein, it has to tumble and rotate. It needs like 6 phosphorylations, but it binds to a protein which has only one hole, so they can't make sense of that threshold. But what happens is, the idea is it creates some kind of electrostatic cloud around it through its motion, which then just reaches the strength to actually bind to the protein. So without activity, you don't have, the structure to interact and all that. So it throws, it overthrows our old picture of like the molecular machine and all that. And they talk about these as, at some point they talked about these proteins as dancing clouds, because you couldn't really see them in an X-ray crystallography, or you would have to look at NMR. You get scattering. Anyway, they would, like, the terminology is really close, and they talk about ensembles of structures rather than the structure. And we saw, like, in the viral cloud literature, you find ensembles of sequences. And so ensemble thinking, again, energetics, I think that's a very powerful way. And then biologists would say, well, but, the protein is really stable. And so I have a paper that will come out in Hühle, the Journal of Philosophy of Chemistry, which talks about what I call energy level management practices, which are like, this goes now into lab ethnography maybe, which I'm not doing professionally, but kind of just interested in. It's kind of all the practices that are constantly there, but are constantly overlooked, like the ice bath, the buffer solution, which are essential to kind of create what you just showed, something that has one defined structure. And obviously that's probably crystallized, so it's more richly defined anyway, but just kind of, there are all these processes that humans put in to make these stabilities, so that then can have reproducible science and, repeat the experiment and kind of have the same thing there after 2 hours on, doing experiments. You don't want it to fragment and all that. So all of these things kind of start creating a molecular picture that is very independent. And so that goes into, again, kind of the basic assumptions of our thinking, overlooking processes and practices, not just the processes of nature, but human intervention as well. And you get this meshwork of different decelerations, because you put it on ice to decelerate certain molecular movements. And so you have to think the whole nexus of, or society of events, which human activities are part of. And I think that's very interesting. So that's why I thought viruses are so interesting, because they are molecular machines, and they really force you to like think like, what's going on here? How can we open up this thinking? So yeah, so I'm just rambling, sorry.
I thought that was beautiful. I would love you to ramble more, please.
Yeah, no, now I'm all out rambled. Yeah, so anyway,
well, maybe one basic question. So, I gave a keynote lecture about kinases and phosphatases at a multi-species studies conference, and everyone just like stared at me blankly after I gave this like hour-long talk. But if you could slow down for folks who haven't taken a cell biology class and just talk about phosphorylation and what that means in this kind of process view of cellular biology, but also how, do you see that, phosphorylation processes, are there viruses that are doing the work of, do they have kinases and phosphatases that are getting in there and changing how some of those dynamics are taking place?
I mean, phosphate, ATP, phosphate, phosphatases, kinases. I mean, this is the signaling. Yeah, I mean, this is what the cell, how impulses are passed on, impacts, and that triggers something. And you have a phosphorylation is a small phosphate group, P phosphate and 3 oxygen. Yeah, PO3 4 minus. I'm not even sure. My God, my biology is going down. PO4 3 minus, PO3 4 minus. Anyway, phosphate group, small group, is transferred to a protein. It's highly charged, so it changes the surface properties, and then it can interact with something else which wasn't before opened because it depends on electrostatic interaction. You can form new complexes. It's a very machine mechanistic view of the cell. I mean, it explains so many things so beautifully. You have two proteins can't interact, another protein adds a phosphate, and suddenly that interaction can happen, and you can trigger downstream interactions and all that. But the narrative, if you look at that literature, you have ATP. Why is it so powerful? Why is it an energy carrier? Because it has like this high energy bond. Anyway, it's very substantialized again. It just carries, the ATP just has this energy. The phosphate just has this effect. But when you dig into the chemistry of it, it's not, ATP doesn't have a powerful bond. Anyway, that goes too much into biochemistry because it depends on the whole reaction system. It's again, all energetics as well. The thing, are viruses, yeah, viruses modulate those systems as well. It's again about, it's a flow of energy that can be embodied in this thing, phosphate or ATP or proteins. But if you look at it more closely, it becomes again an intersection of different processes that intersect with each other's intensity. I don't have a specific example of a virus. Does anyone have one here? I don't have like a specific example of the virus coding for some sort of modulator of other kinase. I'm not sure. There will be many inhibitors that they express and all that. But yeah, phosphates are really just again an energy flow system.
Start or end with my same point. I can't wait for the book. I think this is going to be a fabulous, book-length project. And you've enlightened us about, a lot of things, like taking apart some of the foundational ways that we're thinking about life, thinking about stability of entities, fundamental questions about epistemology and ontology, not only in terms of, reality as it is, but reality as it's sort of co-produced in a lab with, the cold baths slowing down the velocities, intensities, so that we can actually, make images of them, even if the images are fragmented and distorted, even as they're produced. So I feel enlightened today, and yeah, thank you so much for sharing this with us, and can't wait to continue. And eventually we will get to, we're dancing around Whitehead. We haven't actually read any Whitehead yet in this group, but we've got a couple of good texts in the queue. And Rachel, do you want to give a plug for next week? I'm forgetting. I think it's what's on.
Jamie Lorimer.
Jamie Lorimer, right. And I think it's in this exact same time slot, right? The 9 a.m. Central European time?
Yes.
Okay. Jamie Lorimer talking from his new book, The Probiotic Planet. So thank you, Stephan. Let's give you a virtual clap. And
thank you, everyone.
Thanks for coming, everyone. See you next week to talk about probiotics.
Thank you, everyone.
Cheers, and congrats again on the job.
Thank you.